is purple. Now, Mendel had to ask himself, is this a pattern of inheritance? Okay, what do you think? I think he was right. He was onto something. He obviously put one trait at a time, but he was able to distinguish that in the F2 generation, 75% were purple or had the same amount of trait as their parent from the first monohybrid cross, but only 25% came up to be like one of the grandparents, right? So that was curious. What were his hypotheses? He came up with 4 hypotheses. The first one was that there were alleles. And alleles are basically alternative forms of genes that account for variations in inherited traits. So for example, for purple flowers, we get 2 alleles. One coding for purple, one coding for white. Now, if we get them both purple, truly bred, one after the other, then the parent will have this kind of genotype, P. The F1 generation will have this kind of phenotype. And the F2 generation, we get both. You get basically A, and also the other grandparent that I didn't write. You get the point though. So the other parent that I missed was AA. Okay, so we get these for the F2. And we call these genotypes. Okay, sorry about the writing. I'm not really in control of that. But you get the idea that this right here, big letter, is representative of one allele of the dominant. And this one right here is one allele that's recessive. 2 versions of one trait. The second hypothesis he had that all organisms have 2 alleles. Now I know I discussed that here, so I'm pretty sure you know what that means. Basically that we have, either dominant or recessive form of the allele, okay? And if you get, 2 of one form of allele in one offspring, that's called homozygous. Okay, homozygous. The root word here, the prefix is homo, which means, what do you think it means? It means of your own, like your own. Okay. Whereas hetero is the root word for meaning 2. Okay, meaning the other kind. Okay, other. Okay. Oh, look at that cursive. That came out well. So there are 2 forms of alleles, and 2 identical alleles give us homozygous, whereas 2 different alleles give us the other kind, is heterozygous. Now, his third one we already talked about again, because, I'm always so ahead of myself. The 2 allele pairs that are different give us the appearance of a dominant allele. Okay, so AA, in case of our purple, will actually be purple. Even though it has both purple and white flower, it will look purple. Whereas AA for sure, big A, means that it's again purple. But what about little a? That means it's white. Okay, so you see, the more dominant one here wins, which is purple, right? His fourth hypothesis, which is not that complicated, but you've learned this in meiosis, okay? That a gamete has only one allele of that trait. So a gamete has half the amount of chromosomes, okay? So this is an egg, and here's a sperm, has the other half. And these 2 together, each one has the allele of that one trait. So let's say we're trying to determine if this egg and sperm will make purple. What do you think the possibilities of the alleles in the parents? Okay, so one parent will carry the big A allele, and one other parent will carry the allele maybe for little a or big A. Okay, so the combination of big A or little a will give us purple, or the combination of big A and big A will give us purple. Same thing here. But both of them cannot together have a little one or the other A that's little as well. So one allele comes from each. So if we choose this to be the genotype for this phenotype, then that would be correct. On another occasion, this could be the egg carrying the big allele, which is the dominant allele, or the sperm can carry the other dominant allele to give us purple. So there are two scenarios to this case. Okay, and remember these are crossed together to give us 100% or 2/2 of an individual offspring. I really should not write with this. Offspring. Okay. So 2 out of 2 give us what? 1. Come on, guys. It's math. Alright, let's go to the last part of this presentation, which is the chromosomal theory of inheritance. Now, Mendel, he published his results without knowing much about chromosomes, or anything about DNA for that matter. But it wasn't until the 1900s that people started to wonder if Mendel was right. Okay, he called them at the time heritable factors, but they're actually what's known as genes. Okay, so at the time he called them heritable factors because that's what he identified them as, but actually that's what they're commonly known by in our scientific language. So his observations began to gain some ground, and people started believing in them. And so some people started to identify these genes and the alleles, in the way that meiosis separates. So remember meiosis, you have the interphase where DNA, replicate, condenses, and then it goes to, so we'll call that I. And then it goes to what? Prophase, okay? Prophase. And in prophase, basically, we get this crossing over, okay? So you got 2 chromosomes here crossing over, exchanging DNA. And after that, it goes on to metaphase. And in metaphase, after they have crossed over, let me show you how they cross over. Maybe I can use another color. Here, blue. Okay. And let's say, the other one is blue, but then it also has red because it exchanged some of this stuff, right? So these two have exchanged. All right, so from that, we get metaphase. And in metaphase, they separate. There's a metaphase plate, right? And these two separate, beginning to divide. In metaphase, it goes on to anaphase. Basically, you get, what we got here. And then the blue. Okay, and remember, they're pulled by these spindle fibers. We are going to show them in green. Alright. But then, and let me label that anaphase. And then it goes on to telophase, where you get 2 separate cells. Okay, and 2 separate cells look like this. And remember, we have the other blue chromosome. Imagine I copied over all of this exchanged DNA. And that's telophase, basically. Oop, I changed colors. And from telophase, we go into meiosis 2, where it goes through interphase, metaphase, anaphase, telophase. But in the end, these two, each one breaks into 2. So we get 4 daughter cells. And in the first one, could have 1 copy. and 1, but remember it'll have that blue part, right? Okay, and then 1 will have the other chromosome, one of these, and we'll have the red part that's been crossed over. So that's half the number we started out with. If you look at this, you have 4, and then they divide into 2, and then they again divide in half to give us 1 each. So that is a gamete. So people was trying to wonder, where are the alleles in all this? Well, the alleles are actually located on the chromosomes. This A can be here, this A can be here. So let's trace it back. This A will stay here, stay here, stay here, and go there. This A can stay here, stay here, stay here, and then go there. So big A, little a will be here. What about the other 2? Well, I'm glad you asked, because everything that happens on one part is also available on the other, because remember, these are duplicated from interphase. So they'll still have these same genes again over here. Okay, so you get the 4 combinations that are available. And basically, that's how we trace those genes from meiosis. And we can use this information from the gametes for the eggs to each have an allele of a different kind. Alrighty, let's go on to the next slide, which is our last, and summarize our chromosomal theory of inheritance, which is what we just went through over here. Okay, this is the chromosomal theory of inheritance. And basically it told us that, this guy Walter Sutton and Theodore Boveri independently linked Mendel's rules with the details of meiosis to formulate this rule of inheritance. And meiosis caused the pattern of inheritance observed by Mendel in his pea plants, which we call, his hereditary factors to be genes. Remember that allele pairs in our studies of meiosis and mitosis, that each diploid cell has 2 sets of homologous chromosomes. So let's go back here. This is a diploid cell, okay, and this is what we call a homologous pair, okay, because they're paired up and they have the same information, but they're crossed over. It is on these homologous chromosomes that the allele pairs for each trait, which is Mendel's second hypothesis, are found. Let's go to his second hypothesis. Second hypothesis says that all organisms have 2 alleles. He was right. Good job, Mendel. Alright, so that's all we got for here. Let's go back to the vocabulary list. Do you think you learned carpal today? Do you think you learned cross-pollination? What about dominant, F1, F2, fertilization, genotype, heredity? Heredity and inheritance, by the way, mean the same exact thing. So if you know what that is, you'll know what this is. Inheritance is the passing on of traits from parents to offspring. Look it up in your PSI slides, it's there. Hybridization is basically breeding, okay? So you're breeding 2 parents and you get a hybrid offspring. Voila, you just learned that. Phenotype, we know that phenotype means physical trait. Recessive means the less dominant allele. Stamen is the male part that fertilizes the carpel, which is the female part. Stamen, remember stamen, sperm, S and S, male, okay? Trait means the same exact thing as a phenotype. Okay, so if you know this, you know that. And